Good morning, class. My name is Henry, and this is MLS 304. Now, for this class, we'll be talking on immunoassay, and for this level, we'll be dwelling more on radio immunoassay and ELISA techniques. I will have different kind of ELISA techniques. They are direct, indirect, sandwich, and competitive. Now, for you to understand more about these immunoassay techniques, I'll be playing some videos, or I will share from some videos. So stay tuned and listen or watch.
Of Laboratory Medicine series will serve as an introduction to clinical immunoassays. Topics for discussion will include basic antibody structure, immunoassay designs, common types of immunoassays found in the clinical laboratory, and mechanisms of interferences seen with clinical immunoassays. Antibodies are glycoproteins commonly referred to as immunoglobulins and are produced by the immune system when a foreign molecule is detected in the body. Antibodies have constant regions and variable regions, with the antigen binding site located in the variable region. Monoclonal antibodies are produced by a single clone of plasma cells and all bind to the same epitope of an antigen. Polyclonal antibodies are produced by many plasma cell clones and bind to a wide variety of epitopes on an antigen. How tightly an antibody binds to an antigen is termed the affinity, while the number of potential binding sites is the avidity. Analytical methods that use antibodies for detection are referred to as immunoassays. The reasonably specific binding of antibodies to their targets make immunoassays effective methods for detection of analyzing complex biological matrices. Several different types of immunoassays are used clinically and differ in design, detection mechanism, and how the assay reagents are combined with the sample. Heterogeneous immunoassays require separation of the analyte-antibody complex from the remaining sample prior to final analysis. This can be accomplished using precipitating chemicals, cross-linking with other antibodies, or use of an antibody bound to a solid phase. Once the remaining matrix components are washed away, the remaining assay components are added for final detection. Homogeneous assays can distinguish between free and antibody-bound analyte and do not require a physical separation of analyte-antibody complex from the remaining sample. Immunoassays also differ on the type of reaction method used. Competitive immunoassays restrict the number of antigen binding sites, resulting in a competition for antibody binding between the endogenous analyte and a detectable labeled analog. As a result, the amount of labeled analog bound is inversely proportional to the amount of analyte in the sample. As the amount of analyte in the sample increases, the detectable signal decreases. Competitive immunoassays can be classified as simultaneous addition, where all components are added at once, or sequential addition, where the sample is incubated with the antibody before the labeled analog is added. Non-competitive immunoassays are designed to have excess antibody binding sites and produce a signal directly proportional to the amount of analyte in the sample. In sandwich immunoassays, two separate antibodies are used. A capture antibody is bound to a solid support and is used to extract the analyte of interest from the sample. A second antibody is labeled to allow detection and binds to a separate site on the analyte, resulting in an antibody sandwich with the analyte positioned in the middle of the two antibodies. As the amount of analyte in the sample increases, the detectable signal increases. For final detection, a wide variety of immunoassay detection methods are currently used in the clinical laboratory, including fluorescence, chemiluminescence, radioactivity, and enzyme-based detection. Fluorescence detection uses a dye-labeled antibody directed against an analyte of interest with the amount of detected fluorescence proportional to the amount of analyte in the sample. Another common type of fluorescent immunoassay is a fluorescence polarization immunoassay that uses fluorescently labeled analogs rather than a labeled antibody. When labeled analog is unbound by antibody, it is free to rotate and there is low polarization of light. When the labeled analog becomes bound by antibody, its rotation is slowed by the mass of the antibody and there is an increase in polarized light. The change in polarization is directly proportional to the amount of analyte in the sample. The most common enzyme-based antibody labels include alkaline phosphatase, peroxidases, glucose-6-phosphate dehydrogenase, and beta-galactosidase. Enzymatic immunoassays are advantageous due to the amplification inherent to the enzymatic process. The substrate-to-product reaction can produce a colored product, fluorescent product, a photon, or even an intermediate substrate for a second enzymatic reaction. Enzyme-linked immunosorbent assays are common in the clinical laboratory and use a capture antibody and an enzyme-labeled detection antibody. Many automated instruments use an enzyme-labeled detection antibody and a chemiluminescent substrate producing a photon for measurement. Two other types of enzyme detection include EMIT and CEDIA. An enzyme-multiplied immunoassay or
Please hold on for the second video. Hold on for the second video.
In previous video lectures, we have covered immunotechniques based on precipitation and agglutination reactions. There we saw that antigen-antibody reactions results in the formation of large immune complexes, or antigen-antibody complexes. These immune complexes are large enough, and we can observe them by naked eyes. But how to detect the presence of antigen or antibodies in case when antigen-antibody interactions do occur, but they are not visible? This may be because the test antigen or antibody is present in very low concentration. The precipitation and agglutination immunotechniques will not detect individual antigen-antibody interactions. Two most popular techniques are radioimmunoassay and enzyme-linked immunosorbent assay. RIA and ELISA are labeled, or tag immunoassays. Today we will understand, what do we mean by labeled or tag immunoassay? What role does radioisotopes and enzymes have in these assays? Let's begin. Assay means analysis, or simply, a test. The term immune reflects that these tests are related to our immune system and its mechanism of action. Immunoassay can be defined as the technique or test to detect the presence or quantity of substances such as hormones, drugs, and specific proteins based on specific antigen-antibody binding reactions. These substances can act as antigen or antibody. Now, next question is, what do we mean by tag or labeled immunoassay? Suppose this is an antigen immobilized in a microtiter well. This is the specific antibody to the given antigen. We add this antibody to the microtiter well. What will happen? Since this antibody is specific to the given antigen, it binds to the antigen. Now, this binding is not visible to us, but it took place. So, what can we do to confirm that binding has occurred? Again, this is a microtiter well containing the same immobilized antigen. And this is the specific antibody to this antigen. But this time, this antibody is tagged, or labeled. By tagged, or labeled we mean that this antibody is bound covalently to a detectable tag. This tag can be a radioisotope, enzyme, etc. We will discuss them further shortly. Now, when we add this tagged, or labeled antibody to the microtiter well, the antibody will bind its specific antigen as usual. But, this time we can confirm the binding by detecting the tag or label. So, at this point we understand that tag or labeled immunoassay is a test to detect substances that are present in very low concentrations. The test result is observed indirectly. That is by detecting the tag or label bound to either the antigen or antibody partner. Let's understand role of two important tags or labels, which are radioisotopes and enzymes. Radioisotopes are used in radioimmunoassay. We know that an atom is composed of three subatomic particles, namely proton, neutron, and electron. The nucleus of an atom, which is made up of protons and neutrons, together make the mass of the atom. Isotopes are the atoms of the same element that have different number of neutrons in the nucleus. That means isotopes of same element have same atomic number, but different mass number. For example, hydrogen has three isotopes: protium, deuterium, and tritium. Here you can see that no neutron is present in protium. One neutron is present in deuterium, and two neutrons are present in tritium. First two isotopes of hydrogen, that is, protium and deuterium, are stable, but the third isotope, that is tritium, it is unstable. These unstable isotopes emit particles or energy to reach a stable form. They emit radiations such as alpha rays, beta rays, and gamma rays. Therefore, such isotopes are known as radioisotopes. Radioisotopes can be covalently attached to proteins. As we know, all antibodies and most of the antigens are proteins in their chemical nature. These radioisotopes thus easily label them. The radiations emitted by the radioisotope can be detected and measured using specialized instruments such as gamma counter. Most popular radioisotope used as a tag is iodine-125. It has a half-life of 60 days, which is a relatively long half-life. Let's now understand how enzymes result in detectable immunoassays. Enzymes as tag or label are used in enzyme-linked immunosorbent assay. Suppose this is a microtiter well which contains immobilized antigen. This is our antibody specific to the given antigen. We tag or label this antibody by covalently attaching an enzyme to it. Now, what kind of enzyme is this? These enzymes convert a colorless substrate into an easily detectable colored end product. Let's say this is our substrate. We add it to the microtiter well. Enzyme bound to the antibody act on this substrate, and colored end product is produced. Such substrates are known as chromogens. A chromogen is a colorless substrate acted on by the enzyme to produce a colored product. These enzymes are also known as reporter enzymes. This is because they produce a color change when the enzyme reacts with its substrate. This color change tells the observer that reaction has taken place. Most commonly used enzyme labels are horseradish peroxidase and alkaline phosphatase. A large selection of substrates is available for performing ELISA with horseradish peroxidase and alkaline phosphatase. Most commonly used substrate for horseradish peroxidase is O-phenylene diamine dihydrochloride. Yellow-orange end product is produced when the enzyme acts on this substrate. Similarly, for alkaline phosphate, substrate used is p-nitrophenylphosphate, and yellow end product is produced. That's all in today's lecture. In the next video lecture we will study radioimmunoassay. Thank you for watching.
Okay, for the next lecture now, the next video, just hold on.
In the last video lecture we understood what are tag-labeled immunoassays. We also discussed two important tags, radioisotopes and enzymes. The technique in which a radioisotope is used as a tag or label, that is, radioisotope is covalently linked to antigen or antibody for the detection of antigen-antibody complex, is known as radioimmunoassay. Radioimmunoassay was developed in 1959 by endocrinologist Rosalind Yalow and Solomon Burson to measure the circulating levels of important hormone insulin. In 1977, Rosalind Yalow was awarded Nobel Prize for Medicine for the development of radioimmunoassay for peptide hormones. Solomon Burson could not share the award because of his untimely death in 1972. In radioimmunoassay, the antigens, or antibodies, are labeled with radioisotopes. We know that radioisotopes emit radiations. These radiations can be detected and measured using specialized instruments such as gamma counter. Most popular radioisotope used as a tag is iodine-125. Let's first understand the basic principle of this assay. Radioimmunoassay determines the concentration of an antigen in a sample based on the competitive binding between radiolabeled and unlabeled antigen for its specific high-affinity antibody. Suppose this is a microtiter well. A fixed or constant amount of antibodies specific to our test antigen is immobilized in this well, say, 2 antibodies. Next, we add specific radio-labeled antigens to this well. And the amount of these antigens is such that they saturate all the antigen binding sites present in the well. This well is rinsed to remove the unbound radio-labeled antigens. At this point, if we measure the radioactivity of the well, it will be maximum, say 100%. Now in a second microtiter well, again we take the same amount of immobilized antibody. But this time, we add unlabeled antigens along with the previous, fixed amount of radio-labeled antigen. Note that antibodies present in the well has same affinity for both the labeled and unlabeled antigen, since both the antigens are same. Therefore, these antigens will compete with each other for the antigen binding sites. And now, some of the antigen binding sites will be occupied by the unlabeled antigen. Again, after binding takes place, the well is rinsed to remove any unbound antigens. If we now measure the radioactivity of this well, it will not be 100%. It will decrease because the unlabeled antigens have replaced some of the radio-labeled antigens. Decrease in radioactivity tells us that the antigen is present in the test sample. But how to determine the concentration of this antigen? Suppose we want to detect presence and quantity of an antigen in the patient's serum. We suspect that this patient's serum has the antigen of interest. To quantitate the antigen present in the patient's serum, first we need to obtain a standard curve. This standard curve is obtained by measuring the amount of radioactivity using fixed and excess concentration of radio-labeled antigen and varying known concentrations of the unlabeled antigen. Again, note that both these radio-labeled and labeled antigens are same as the antigen we are going to detect and quantitate in the patient's serum. Let's have a look at the basic steps involved in the generation of standard curve. We require 3 main components. An important point is that all these components, and their quantity is known to us. First component is specific antibody to the test antigen. The quantity of this antibody being used is fixed. These antibodies are immobilized in microtiter wells. Second component is the radio-labeled antigen. Asterisk symbol used in this designation tells that the antigen is labeled. The quantity of radio-labeled antigen is again fixed. This quantity is enough to saturate all the antigen binding sites in the well. Third component is the unlabeled antigen. We will use increasing varying concentrations of unlabeled antigen. First, we take a microtiter plate. To keep this illustration simple, we are showing only 5 microtiter wells. In each well we will immobilize fixed amount of antibodies, specific to the test antigen. Let's say 3 antibodies are fixed in each microtiter well. Thus, concentration of antibody is constant in all these wells. Now, we add fixed amount of radio-labeled antigen to the first well. This amount of radio-labeled antigen is enough to saturate all the antigen binding sites in the microtiter well. We will rinse the well to remove any unbound antigens. If we measure the radioactivity of this well, it will be maximum. Let's say 100%. This is our control well. Next, we will add same fixed amount of radio-labeled antigen in all other wells too. But along with this, we will add unlabeled antigen in increasing varying concentrations. That means, to each well, fixed amount of radio-labeled antigen and increasing amount of unlabeled antigen is added. What will happen now? As we said before, antibodies present in the wells cannot distinguish between radio-labeled and unlabeled antigens. Thus, in each case, the unlabeled antigen will compete with the radio-labeled antigen for the antigen binding sites. It is important to note that, as the concentration of unlabeled antigen increases, it occupies more antigen binding sites. Now, in each well there will be bound as well as unbound antigens. In the next step, we will separate the unbound antigens by rinsing the wells and measure the radioactivity in each well by gamma counter. Radioactivity will decrease in each well with increasing amount of unlabeled antigen. This is because now some of the antigen binding sites have been occupied by unlabeled antigens, and there is less number of bound radio-labeled antigens in each of these wells, as compared to the control well. At the end of this procedure, we have value of radioactivity for each well. Also we know the concentration of the unlabeled antigen added in each well. Let's now plot a standard curve. In this graph, y-axis represents the radioactivity measured for each well, and x-axis represents the concentration of the unlabeled antigen. As the concentration of unlabeled antigen increases, the value of radioactivity decreases. So, this is our standard curve. Note that we are showing only the linear portion of the curve. Let's now quantitate the antigen in the patient's serum. We will take same quantity of immobilized antibodies in a microtiter well, as we used in the standard curve. To this well, we will add a fixed amount of radio-labeled antigen. Again, this amount is same as we used in standard curve. And along with this, we will add suspension of patient's serum. The antigens present in the patient's serum will compete with radio-labeled antigens for antigen binding sites in the well. Well is rinsed to remove unbound antigens, and radioactivity is measured. Let's suppose radioactivity measured for this well is 35%. Decrease in radioactivity indicates the presence of antigen in the patient's serum. We will extrapolate this value of radioactivity in the standard curve. Thus, we will get to know the quantity of antigen present in the patient's serum. Radioimmunoassay is mainly used to detect hormones and to diagnose allergies. These tests are now replaced by more sensitive and cost-effective methods. The main disadvantage of using radioimmunoassay is the hazard to the professional due to exposure to radioactivity. Most of the radioisotopes have short shelf life. This technique is also expensive as it requires specialized equipment. Moreover, it is a time-consuming technique. That's all in today's video lecture. In next video we will study enzyme-linked immunosorbent assay. Thank you for watching.
Now for the next video on ELISA.
ELISA stands for enzyme-linked immunosorbent assay. ELISA is a labeled immunoassay where the antigen or antibody is labeled with a reporter enzyme. Recall that reporter enzymes produce a color change when the enzyme reacts with its specific substrate. Thus in ELISA, color change is used to identify a substance. This assay is used for the detection and quantitation of substances, such as peptides, proteins, antibodies, and hormones. There are three main formats of ELISA. These are direct ELISA, indirect ELISA, and sandwich ELISA. In this video lecture we will study direct ELISA. In the previous video lecture, we understood the difference between the term direct and indirect detection assay. Easiest way to remember the difference between direct and indirect ELISA is to look at the enzyme-linked antibody. If enzyme-linked antibody is directly binding the antigen, it is direct ELISA. And if enzyme-linked antibody is not directly binding to the antigen, then it is indirect ELISA. As you can see here, in indirect ELISA, the enzyme-linked antibody binds to the antibody already bound to the antigen. Let's now understand the steps involved in direct ELISA. Direct ELISA is used to detect antigens in the test samples. Test result can be positive or negative depending on the presence and absence of the antigen in the test sample. Suppose we have serum samples from two patients, patient 1 and patient 2. For our illustration, we assume that antigen is present in patient 1 and absent in patient 2. Let's see how to detect and confirm this by ELISA test. We will take two microtiter wells, each corresponding to the patient's serum sample. So this is 1, and this is 2. In the first step, the antigens present in the patient's serum are adsorbed and immobilized in the microtiter well. No adsorbed antigens are shown in the well 2, because the target antigen is absent in the serum sample of patient 2. Note that we are not showing other antigens present in the serum of patients. Both these microtiter wells are rinsed to remove any unbound antigens present in the well. In the second step, enzyme-linked antibodies specific to the target antigens are added in each well. These enzyme-linked antibodies will bind to their specific antigens in the well, if they are present. Thus, here you can see that antigen-antibody binding takes place in the first well, and no antigen-antibody binding takes place in the second well. This is because there was no target antigen. Each well is rinsed again to remove any unbound antibodies. After rinsing step, only antibodies present in the first well are those bound to the antigen, whereas no antibodies are present in the second well. In the third and final step, we add substrate specific for the enzyme that is linked to the conjugated antibody. After some time, color development can be seen in the first well. This is because the enzyme linked to the antibody acted on the substrate and resulted in the formation of colored end product. But in the second well there is no color change. This is because there was no enzyme-linked antibody present in this well. Thus, in ELISA test, a color change reveals the presence of the antigen. This is termed as a positive test. No color change indicates that the antigen was not present in the test sample. This is termed as a negative test. Thus, direct ELISA detects the presence of antigens in a test sample. It is commonly used for immunohistochemical staining of tissues and cells. That's all in today's lecture. In the next lecture we will study indirect ELISA. Thank you for watching.
Alright, indirect ELISA. Indirect ELISA. The next video.
In the last video lecture we studied direct ELISA. There we saw that enzyme-conjugated antibody directly binds the target antigen. And when substrate for the enzyme is added in the well, color development indicates that the target antigen is present in the sample. Let's now study indirect ELISA. Indirect ELISA is the most popular. It is used for the detection of specific antibodies in a serum sample. For example, for antibodies to HIV, rubella virus, and to detect certain drugs in serum. Suppose we want to detect and confirm the presence of particular antibodies in the serum of the patients. These are two serum samples from two different patients. Let's assume antibodies of interest are present in serum of patient 1 and absent in serum of patient 2. Other antibodies may also be present in the serum of these patients, but we are not showing them here. We will take two microtiter wells. In the first step, specific antigens to the antibodies of interest are immobilized in both the microtiter wells. This means these antigens are known to us. For example, if we want to detect antibodies for HIV, the antigens absorbed in microtiter wells are recombinant envelope and core proteins of HIV. After some time, these wells are rinsed to remove any unbound antigen. In the second step, serum samples from each patient is added in their respective wells. Since antibody of interest is present in the serum sample of patient 1, these antibodies will bind to the specific antigen present in the well. But no antigen-antibody binding will take place in well 2 because antibodies specific to the antigen are absent in serum sample of patient 2. Also, other non-specific antibodies present in these serum samples will remain unbound. Rinsing is done again to remove unbound and non-specific antibodies from the well. Here, we know that the antibody binding directly to the antigen is the primary antibody. In the third step, enzyme-linked secondary antibodies are added in each well. Recall that these secondary antibodies have antigen binding sites for the Fc regions of the primary antibody. Thus, they bind to the primary antibody present in the first well. Since no primary antibody is present in the second well, the secondary antibodies added in this well will remain free or unbound. Again, wells are rinsed to remove any unbound secondary antibodies. In the fourth and final step, substrate specific for the enzyme linked to the secondary antibody is added in each well. In well 1, since enzyme is present, it acts on the substrate, and color development can be seen. This is a positive test. In well 2, no enzyme-linked antibody is present. There is no enzyme to act on the substrate, and hence, no color development. This is a negative test. So, this was indirect ELISA. Let's quickly recall and compare direct and indirect ELISA. Direct ELISA is used for the detection of antigens. Indirect ELISA is used for the detection of antibodies. In direct ELISA, enzyme is conjugated to the primary antibody. In indirect ELISA, enzyme is conjugated to the secondary antibody. In the next video lecture, we will study sandwich ELISA. Thank you for watching. We have covered direct and indirect ELISA. I hope you now understand the basic difference between these two types of ELISA. Today we will study most commonly used format of ELISA, which is sandwich ELISA. Sandwich ELISA is used for the detection of antigens. Most popular application of sandwich ELISA is point-of-care testing and home test kits such as home pregnancy test kits. Sandwich ELISA uses two different antibodies for the same target antigen. Important point is that these antibodies recognize and bind to different epitopes on the target antigen. Let's understand the details of sandwich ELISA. Sandwich ELISA requires three main components: capture antibody, target or test antigen, and an enzyme-linked antibody. Thus, in sandwich ELISA, an antigen is trapped between two antibodies. In the first step of sandwich ELISA, capture antibody is immobilized in the microtiter well. Capture antibody is a high-affinity antibody, specific to the target antigen. Its function is to capture the target antigen from the test or serum sample. So, after the immobilization of capture antibodies, the microtiter well is rinsed. This is done to remove unbound antibodies present in the well. In the second step, serum sample, or test sample, is added in the well. Now, serum from a patient may contain many different antigens, but the capture antibody is specific for a single target antigen. If the sample contains the target antigens, they will bind to the capture antibody present in the well. If the specific antigen is absent in the test sample, no antigen-antibody binding takes place. Again, the well is rinsed to remove unbound and nonspecific antigens. In the third step, an enzyme-linked antibody is added in the well. The enzyme-linked antibody binds to the antigen. Recall that these antibodies binds to the epitopes, different from those bound by the capture antibody. After some time, well is rinsed to remove unbound enzyme-linked antibodies. In the fourth and final step, the substrate specific for the enzyme linked to the antibody is added. Color development confirms the presence of the antigen in the test sample. Sandwich ELISA can be further divided into two types: direct sandwich ELISA and indirect sandwich ELISA. This classification is based on whether the enzyme-linked antibody is directly or indirectly detecting the antigen. Here, the target antigen is being detected by the conjugated antibody itself, so this is direct detection. But here, a primary antibody is first binding to the target antigen, and the conjugated secondary antibody is detecting the antigen-bound primary antibody. So, this is indirect detection. That's all in today's lecture. In the next video lecture, we will understand.
Hello guys, in the next lecture, next video, we're talking about competitive ELISA. So stay tuned.
Hello friends, today we are going to discuss about competitive ELISA. Competitive ELISA is used to determine the concentration of either antigen or antibodies present in a particular sample. Let us discuss how this particular technique is used to determine the concentration of antibody. Suppose we have to determine the concentration of anti-A antibodies from a serum sample. That is, the antibodies which specifically binds to the A antigen. For this, we will require A antigen in this case. Means you must have the antigens specific to the antibodies whose concentration is to be determined. For example, if you want to determine the concentration of anti-HIV antibodies from the sample, you must have HIV antigen with you. Second and the most important thing that you must have is the enzyme-linked anti-A antibody. This antibody, it also binds to the A antigen like the antibodies which are present in the sample. But these antibodies, they are coupled to the enzyme. The enzyme can be horseradish peroxidase, beta-galactosidase, or alkaline phosphatase. All these enzymes have chromogenic substrates. For example, TMB, that is tetramethylbenzidine, which is the substrate of enzyme horseradish peroxidase, gives blue color when converted into the product, and its intensity can be determined by spectrophotometric method. In the first step, a microtiter well is coated with A antigen like this. The coating is mediated with the help of coating buffer. After this, the sample containing anti-A antibodies whose concentration is to be determined and a fixed amount of enzyme-linked anti-A antibodies are added to the well. These two types of antibodies will compete with each other to bind the antigen present in the well. Now you might think, which type of antibody is going to win this competition? Well, the antibody with the greater concentration wins the competition. Now we don't know which kind of antibody's concentration is more. Let us assume both the situations. If the concentration of antibodies from the sample is more than enzyme-linked antibodies, as in this case, these antibodies will occupy most of the antigens of the well. On the other hand, very few enzyme-linked antibodies will bind to the antigens. But if the condition is reversed, as in this case, most of the antigens will be occupied by the enzyme-linked antibodies. After this, the well is washed to remove unbound antibodies. This step is followed by addition of chromogenic substrate and incubation for a fixed period of time. After incubation, the reaction is stopped by adding strong acids. Now during this incubation, the enzyme connected to the enzyme-linked antibody will convert this substrate into the color product. Now if the number of enzyme-linked antibodies are less in the well, as in this case, less product will form, and hence the color intensity and its absorbance value will be less. And if the number of enzyme-linked antibodies are more in the well, as in the second case, more product will form and therefore absorbance value will also be more. So in short, if the absorbance of the product is less, concentration of antibodies in the sample is more. And if the absorbance is more, the concentration of the antibodies in the sample is less. The actual concentration of antibodies present in the sample can be determined by extrapolating the value of absorbance on the standard graph, like this. So in this way, the competitive ELISA is used to determine the concentration of antibodies. Exactly reverse method is used to determine the concentration of antigens.
Hello guys, I hope you guys understand those videos. So now for the next video, I will show you guys how to understand how to carry out practical ELISA in the laboratory. So sit tight and watch.
Edvotech instructional videos. The enzyme-linked immunosorbent assay, or ELISA. For this experiment, you will need a microtiter plate, phosphate buffered saline, antibody and antigen samples, a color-changing substrate solution, a 37-degree incubator, an adjustable micropipette and tips, and transfer pipettes. Step 1. Label the microtiter plate and transfer pipettes as noted in the product literature. Step 2. Add 100 microliters of the antigen solution to all of the wells. Step 3. Incubate for 5 minutes at room temperature. At this time, the antigens will non-specifically adhere to the plastic through hydrophobic and electrostatic interactions. Step 4. Remove all the liquid from the wells using a transfer pipette. Step 5. Wash the wells by adding approximately 8 drops of PBS buffer to each well. Remove all the PBS from each of the wells, taking care to prevent the buffer from spilling over into adjacent wells. Traditionally following this step, the wells are blocked with a protein-containing buffer to prevent non-specific interactions between the antibody and the plastic wells. We have optimized this experiment to eliminate this step. Step 6. Add reagents as outlined in the product literature. Remember to use a clean micropipette tip for each reagent. Step 7. Incubate the plate for 15 minutes at 37 degrees Celsius. Step 8. Remove the liquid from the wells using the appropriately labeled transfer pipette. Step 9. Wash each well with fresh PBS. Remove the liquid using the transfer pipette designated for each sample. Step 10. Add 100 microliters of the secondary antibody to each of the wells. Step 11. Incubate for 15 minutes at 37 degrees Celsius. Step 12. While the samples are incubating, prepare the detection substrate as specified in the product literature. Step 13. Remove the secondary antibody solution from the wells using the transfer pipette designated for each sample. Wash each well once with fresh PBS buffer. Remove the liquid using the transfer pipette designated for each sample. Step 14. Add 100 microliters of the substrate solution to each well. Step 15. Incubate at 37 degrees Celsius for 5 minutes. Step 16. Examine your results. Differences between negative and positive samples will be obvious, with most positive samples turning brown in color. If color is not fully developed after 5 minutes, incubate at 37 degrees Celsius for a longer period of time.
Welcome back. So I hope you guys understand the lecture. So if you have any question, please do ask or comment on my section. I said comment. Comment on my what's called my YouTube channel. Don't worry, I'll answer all your questions or on the group chat soon. Thank you very much.